Alrighty, so here we are going to talk about a lot of the things in an environment that can impact how enzymes function, mostly like the rate of reaction and how that will be impacted. So, the biggest impact is going to be anything that causes an enzyme to be denatured. So, proteins in general can be denatured. This is when their shape is changed by some factor, usually in their environment. And when their shape changes, they are going to lose their function. So, what you see right here is this is the normal active site of a protein after it's been denatured. The active site is changed, the structure of the protein is changed, and it will no longer fit with the substrate. So there are a number of factors that we'll talk about in a little more detail that can denature a protein and specifically denature enzymes. That is a change in temperature. Usually a higher temperature is going to be what denatures a protein. pH and the chemical environment, so things that are in the environment, various chemicals. In rare cases, not super often, you can have a protein that's been denatured become renatured. In this case, the denaturation would be reversible and the enzyme could regain activity. I am going to say, though, that this is rarer. Usually it would require whatever factor ended up causing the denaturation to be reversed, but then it's also rarer because proteins have to fold in a very specific way. Let's say you changed the pH of the environment of a protein and then you restored the pH back to the original. You would need the protein to fold back into the exact correct configuration, and proteins don't always spontaneously do that. There are sometimes helper molecules that are going to help them fold, and if those aren't present, then you could have the protein misfold and then not only have altered function, but in some case have like a function that harms the cell. So, we're going to talk about the optimal range of some of these traits that are environmental factors we were talking about. Optimal means best or ideal. So, the optimal range is going to be that in which the enzyme is going to function best. So, what affects this? Well, at higher temperatures, you end up disrupting the weaker hydrogen bonds that hold the secondary and tertiary structure in place, and then that can denature an enzyme and alter, usually by decreasing the efficiency of catalysis or how well it helps those chemical reactions happen. pHs outside of the optimal range can disrupt the formation of hydrogen bonds, which can again change the structure, which then usually decreases the catalytic efficiency, meaning that the protein is not going to function as well. So here's what we see. You have something that forms in tertiary structure called a salt bridge, usually between the ionic side chains of a couple of amino acids. So here we have a glutamic acid and a lysine, and notice that they have these charges which then cause them to have that electrical attraction. Well, what you'll see is that when you have an acid or a base present, notice that this is changed where, and if you have hydrochloric acid, say, being added, or you have these ions that have broken off, the chloride ion and the hydrogen ion, those end up attaching to those acidic or basic side chains, and notice they are no longer attracted. So here you have the positive and the negative. Here you have, instead of having, you have a hydrogen bound there, which then cancels out what was a negative charge. And now you have a chloride ion that's attached, and that is being canceled out by the positive charge that was on that. So that changes the 3D structure of the protein, which can affect its activity. So let's look at how temperature affects the enzyme activity. So in general, as you increase temperature, that's going to increase the average speed of the molecules that are moving around, which means that enzymes and substrates are going to hit each other more often, which then increases the rate of reaction. This happens until the optimal temperature is reached. So you'll see this on graphs that show the rate of the reaction. Sometimes it'll be like an actual measurement of the rate at which substrates are used up or products are produced. Other times it'll just be listed as % of maximum activity. And then here what you see is the optimal rate is where you see the peak in this enzyme activity, which looks like it's falling somewhere around maybe 48 degrees. So for this enzyme, 48 degrees is the optimal temperature where you'll have the highest rate of reaction. Notice what happens after that, above the optimal temperature. A lot of times the enzyme will denature, and so the enzyme function will decrease. You have two y-axes on this graph. The rate of reaction is what's being graphed in blue, and then here on the right-hand side, it's the percentage of the enzyme that's actually active. So notice it's very constant up until you hit this temperature of about 47, 48 degrees, after which you start having a sharp decrease in the percentage of the enzyme that's active because more and more of the enzymes are denaturing. Then you can also take a look at how pH affects enzyme activity, because the tertiary structure of a protein is very sensitive to the hydrogen ion concentration, which is again the pH. So enzymes will have an optimal range of pH for their activity. So for instance, a lot of the enzymes in the human body will have their optimal range somewhere between a pH of 6 and 8, because that's the pH of the environment of the cell. So you see here this enzyme trypsin has an optimal pH of what looks like just shy of 9, or here it says pH of 8. Urease, which is another enzyme, is going to have its optimal pH at about 7. But then you have other enzymes like, for example, pepsin didn't show up, my bad. Pepsin here is an enzyme that's found in the stomach, and it is in an environment that's normally very acidic. So its optimal pH is going to be a pH of 2, which is the usual pH in the stomach. If the pepsin ends up leaving the stomach, let's say there's like a tear in the stomach lining or something, once the pepsin gets to a more basic pH, a higher pH, it will denature and no longer be able to break down proteins, which is its normal job. All right. Another factor that can affect enzyme activity is going to be enzyme concentration, which if you have done the catalase lab in class, that is what we were testing as our variable. So the more enzyme you have, the higher the reaction rate, because with more enzymes, you're going to have more frequent collisions with the substrate. So here what you see, this molecule, the red one, that's the enzyme. And then this guy over here, that's the substrate. So then the reaction rate will level off at some point, because if you're not increasing the amount of substrate at the same time, eventually you'll have all of the substrate molecules will be broken down or changed by the enzyme. And so that's going to mean your reaction rate is going to level off, which is what you're seeing over here. The enzyme rate, reaction rate leveling off, because you've basically been, breaking down or building up whatever, all of your substrates. So this amount of substrate then will become the limiting factor to limit how fast this reaction can happen. So if we look, notice I'm increasing the concentration of enzymes, but eventually at a certain point, I have occupied all my substrate molecules have basically bound to an enzyme. And so no matter how much I increase the enzyme concentration after that point, like at this point on the graph, it's not going to increase the reaction rate, because the substrate's all being used up. So some cells, in certain cases, what they can do is they can control the amount of enzyme that's produced. And by that, they can do that by either, sorry, my cat decided to say hi, synthesizing enzyme, or not choosing to synthesize an enzyme. And we'll talk about that when we talk about protein synthesis later. Similarly, substrate concentration can also have an impact. So as you can see in this graph, for a while, as you increase the substrate concentration, the reaction rate will increase as well. Because the more substrate you have, the more often you'll have collisions with an enzyme. But when all the enzymes have their active sites engaged, it doesn't matter how much you increase the substrate concentration past that, eventually it's going to level off because all your enzymes are engaged. That's if you're keeping the amount of enzyme constant. So again, the red molecules here are enzymes. The orange molecules, when they show up, are going to be substrate. And so once the enzymes are saturated, you've maxed out your rate of reaction. So notice I have these substrates popping up. They're occupying the active site of the enzyme. That's going to increase the rate of reaction. But once all the enzymes are occupied, I can keep increasing the amount of substrate, and it's not going to make a difference in the reaction rate because I don't have more enzymes to increase the reaction rate. Now, you can also have molecules that shut down an enzyme's activity. These are called inhibitors. So they can bind to an enzyme in one of a couple of ways that I'll talk about in a moment, and they can slow down the reaction rate. This is also used to regulate metabolism in some cases. So there are two major categories of inhibitors we're going to talk about. Competitive inhibitors bind to the active site of the enzyme. So they physically block the substrate from binding to the active site. Non-competitive inhibitors bind to a different site, and we call those different sites that are not the active site an allosteric site. And a lot of times what will happen is when a non-competitive inhibitor binds, it changes the shape of the active site so it no longer fits with the substrate, and then the enzyme can no longer be active. So when you're looking at competitive versus non-competitive inhibitors, on this graph, what you see is we have increasing substrate concentration as you move to the right. And then on the y-axis, you have the reaction rate. So the green is your baseline reaction where you have an uninhibited reaction. Remember, as you increase substrate concentration, eventually the rate of the reaction will level off because you've occupied all of your enzymes. If you have a non-competitive inhibitor, if you keep the amount of non-competitive inhibitor constant, and you just increase the substrate concentration, the non-competitive inhibitor is going to have an impact no matter how high the amount of substrate you have. A competitive inhibitor, on the other hand, as a competitive inhibitor, so you have your enzyme, and let's say this is our enzyme. This is our inhibitor. This is our substrate. As you have more and more substrate, that substrate is going to, because remember the inhibitor reversibly binds, so it might pop into the active site and then pop back out. Once it's popped out, if there's enough substrate lying around, then the substrate can bind to the active site before another inhibitor can pop on. So as you increase the concentration of the substrate, the reaction rate will increase. So that's one way you can tell if you have a competitive inhibitor or a non-competitive inhibitor. There's also irreversible inhibition when something covalently binds to the enzyme and changes its function. And that can permanently inactivate an enzyme. So some examples, cyanide is a poison, never take cyanide. And what it does is it irreversibly binds to some of the enzymes required for making ATP, and that's how it can kill you. You can't break that cyanide off. And so the only way to get functioning enzyme again is to make brand new proteins. In the meantime, you don't have ATP production, so you've got less energy, which means the chances of the cell dying are higher. A lot of heavy metals can also irreversibly bind with enzymes as well. And then one more way to change the activity of an enzyme is going to be phosphorylation, which is adding an inorganic phosphate, this PI. Usually that's going to happen with ATP providing that phosphate. And this is a way that, frankly, a lot of proteins are turned on and off by either phosphorylating them or dephosphorylating them. And this can basically change like what you see in this diagram here. This is the segment of the protein that's dependent on binding a phosphate. When you bind a phosphate like here, it changes the conformation of the protein because it's changing a hydrophobic region potentially to a hydrophilic one. That can change the 3D shape of the enzyme and either make the active site exposed or bind to the substrate, or it can do the opposite and change the shape of the active site so it can't bind to the substrate anymore. This phosphorylation is done by a specific class of enzymes called protein kinases, and the opposite or dephosphorylating a protein or enzyme is going to be done by protein phosphatases. Again, those aces mean that they're enzymes. All right, so Sorry, I don't have a new source of Bitmoji, so that's just supposed to be my face as part of an enzyme. Hope this was helpful, especially as you're doing the enzymes and cellular regulation sheet.